Hello, everyone. This is Adeline Siu, editor of Pharmaceutical Technology Europe. Joining me today is Dr. Detlef Harp, Director of Research and Development at Hermes Pharma. Detlef, many thanks for being here to speak with us about effervescent dosage forms.
You're welcome.
Firstly, can you tell us what are the advantages of effervescent dosage forms?
Yes, of course. Effervescent tablets are designed to dissolve quickly in water, normally within 2 minutes. Marketing people are very keen on this issue. Effervescent tablets contain edible acids like citric acid or tartaric acid and basic components like sodium carbonates or calcium carbonate. The chemical reaction that takes place is similar to what happens when you bake bread. In baking, you use acid components such as sourdough and baking powder, that is sodium hydrogen carbonate. With heat and some other liquids and solid components, a chemical reaction takes place during the baking process in the oven. The carbon dioxide that is generated leads to a fluffy consistence of the bread. The same is true for the reaction of effervescent tablets. By putting the tablet in water, a chemical reaction of the components starts and carbon dioxide is generated. A patient or consumer can then drink a tasty, lemon-flavored carbonated drink, like a lemonade. Effervescent dosage forms are much easier to swallow than conventional dosage forms, like tablets or capsules, because they are already dissolved in water. About 25% of normal healthy people have difficulties in swallowing tablets or capsules. This percentage increases dramatically with age. The elderly especially have some problems with ingesting tablets or capsules. A big advantage of effervescent tablets is faster onset of action. That's why analgesics are widespread in the formulation into effervescent tablets, because patients want and expect a fast pain relief. This was actually a learning process that started with aspirin and Alka-Seltzer. Studies are available today that show that there is a placebo effect with effervescent tablets. The dissolution of an effervescent tablet sold with aspirin leads to faster pain relief. In fact, it has been scientifically proven that effervescent tablets lead to a quicker onset of action. For example, the maximum plasma concentration of paracetamol is reached 18 minutes earlier with an effervescent tablet compared to a plain tablet. There are several reasons for the faster onset of action. For example, there's a higher pH value in the stomach due to the buffered effervescent solution, and thus a quicker passage through the stomach and the intestine. The user-friendliness of effervescent tablets results from good product taste, which leads to higher convenience, and at the end of the day, better compliance. Larger dose of APIs, for example paracetamol with 1 gram, can easily be digested by patients. Side effects like gastric irritation can be reduced because of these kinds of buffered solutions. The buffered effervescent solution for ranitidine or other actives against heartburn leads to faster effect also.
So, how are effervescent dosage forms produced? Can you tell us about the manufacturing methods and how can you produce high-quality effervescent tablets?
Sure. There are several methods available for manufacturing effervescent dosage forms, with a variety of pros and cons. The easiest way is direct compression, which might be the best option for low-price products, especially food supplements for the mass market. However, usually special pre-treated excipients need to be purchased, which can diminish the price advantage. In addition, usually sugar alcohols are necessary for the mechanical stability, which can negatively impact bioavailability. Direct compression is not suitable for most pharmaceutical applications. For this reason, the components need to be granulated before tableting. There is some interesting research ongoing in dry granulation for effervescent dosage forms, including the compaction of acidic and alkaline components together. The resulting tablets have disadvantages with regard to mechanical and chemical stability, but for stable APIs speaks for an attractive and cost-effective method. Fluid bed granulation is often used for the production of effervescent granules. Usually organic solvents are needed, but they involve higher safety requirements in manufacturing and produce negative environmental impacts. Unfortunately, this leads to higher cost and higher energy consumption. So for new effervescent tablet developments, we at Hermes Pharma recommend using other technologies for producing effervescent dosage forms. Each of these methods has some special advantages. But for all the so-called standard technologies, moisture must be avoided, even in traces. That's why all effervescent producers that use these technologies have to pay high attention to humidity. Sometimes less than 15% relative humidity is required, which results in high costs and negative physiological effects on employees who are working in production facilities. Vacuum granulation is another method suitable to produce effervescent tablets. It allows a very controlled granulation of the products. Maybe we can discuss the advantages of this technology later when we talk about the topo granulation technology that Hermes Pharma uses.
Sounds interesting. So, can you tell us about this topo vacuum granulation? How is the process and what is the difference from conventional methods that are used?
The topo technology is a special patented vacuum granulation technology we use at Hermes Pharma. It is based on a surface modification of the effervescent components, especially citric acid. By adding water, the surface of the citric acid is converted into citrates when using sodium salts into sodium citrates. The citric acid is passivated in this step into citrates. Depending on the duration of the granulation, the conversion rate of citric acid is variable. To control the reaction with water, a vacuum is needed. The proportion of the citric acid converted into citrates can be identified with near-infrared spectroscopy, NIR. The spectral changes due to the conversion of citric acid into citrates can be monitored during the process. The formation of citrates can be detected due to reduced signal intensity of citric acid by NIR inline. A PAT solution for the process was developed in a proof of concept at Hermes. PAT means Process Analytical Technology. Inline analytical instruments, like the NIR device, are used to measure parameters that have been defined as critical for the process. The advantages of the topo technology include the formation of highly stable granules. Manufacturing can take place at higher relative humidity than the other manufacturing methods I described in your previous question. Up to 30% relative humidity is acceptable in the production area. In addition, almost no organic solvents are used. The product possesses high stability against acids and bases due to the passivation of the citric acid and the carbonates on the surface. The resulting effervescent dosage forms created in this way are moisture resistant and have a long shelf life, even in tropical zones. We have proved that the topo technology is excellent, even for several APIs combined in one tablet. Many pharma companies have come to Hermes Pharma with stability issues in their formulations developed with the conventional production processes. For sensitive APIs, the advantages of our technology can be seen easily. For example, in accelerated conditions at 90 degrees Celsius, the tablets manufactured conventionally become yellow or brownish, or unsightly particles appear and stability is compromised, while the ones manufactured with the topo technology remain white. More importantly, no reaction of the effervescent components in the package occurs, even at high temperatures of 90 degrees Celsius. In the strip foils, you can see the difference by measuring the thickness of the packaging after heating. For an effervescent tablet produced with conventional technologies, puffing within the packaging shows up due to the generation of carbon dioxide caused by a reaction of the effervescent couple with traces of water. For an effervescent tablet produced with the topo technology, the strip foil remains completely unchanged. No reaction can take place because of the passivation of the effervescent components. I'm convinced that those examples clearly demonstrate that effervescent tablets manufacturing with the Topo technology are more stable and have a long shelf life, even in tropical zones. Thank you very much for your interest.
Many thanks once again for your insights. It has been a really interesting podcast. This is Adeline Siu, editor of Pharmaceutical Technology Europe.